Hello everyone, I am Dr. Sirajul Faisi. I welcome you in my YouTube channel. Today in this video, I will talk about amino acid and I will start from the beginning. And this video will be beneficial for all competitive exam candidates like IIT, NIT and JEE, for medicals, PSc, MSc students. So let us start. You know what is the functions of amino acid? A variety of roles in metabolism. First, the building blocks of proteins, forming parts of coenzyme as precursor for the biosynthesis of molecules such as heme. Standard and non-standard amino acid. More than 300 different amino acids have been described in nature. Standard alpha amino acid, only 20 are commonly found as constituents of proteins. Non-standard amino acid derivative found in protein, and non-protein amino acid. So why are amino acids uniquely suited for their role as the building block of protein? So first you look at the fundamental structure, general structure of alpha amino acid. This is very important to understanding the protein. So amino group you can see here, carboxyl group you can see here, you can see here R side chain, and R group are reminder of the molecules, and one hydrogens and this alpha carbon. So stereochemistry of amino acid, you can see here for all the common amino acid except glycine. Just remember glycine, the alpha carbon is bonded to 4 different groups. So the alpha carbon atom is thus a chiral center. Now you know chiral from Greek meaning hand. An object or system cannot be superimposed on it, mirror image. So one hand does not match the other when superimposed. Chirality, an object that cannot be superimposed on its mirror image, is called chiral. So mirror, chiral object, non-superimposable mirror image, mirror, non-chiral object, superimposable mirror image. Now chiral molecule, if molecule has an atom bonded to 4 different groups, it can be chiral. Enantiomers, what does it mean? A chiral molecule has left and right-handed isomers called enantiomers. When 2 enantiomers of each amino acids are designated by DL system according to the DL glyceraldehyde, dextrorotation and levo-rotations, L-alanine and D-alanine. Only the L-amino acid have been found in protein. D isomers have been found only in a small peptide of bacteria, cell wall, or in some peptide antibody. So classification of 20 standard amino acid, very important. The name and abbreviation of amino acid. All the amino acids were given a trivial common name. So each amino acid is given 3-letter abbreviation and 1-letter symbol. So they should be remembered. You can see 3-letter abbreviation and 1-letter abbreviation. Classification of amino acid based on polarity, non-polar, aliphatic amino acid, polar, uncharged amino acid, aromatic amino acid, acidic amino acid, and basic amino acid. So non-polar aliphatic amino acid, you can see polar uncharged amino acid, hydroxyl group, sulfur atom, amide group. SH group of the two cysteine protein can be oxidized to form a covalent disulfide bond. Disulfide bonds play a special role in the structure of many proteins by forming covalent link. Aromatic amino acid. They are jointly responsible for the light absorptions of proteins at 280. Protein in solution absorb UV light with absorbance maximum at 280 nanometer. Measuring protein content by photospectrometry. Acidic amino acid, basic amino acid, and all 20 amino acid, 90 of the 20 alpha amino acid primary for proline. It is a secondary amino group acid. Except glycine, the alpha carbon for 19 of them are asymmetric of carbon. So non-polar amino acid, polar uncharged amino acid, acidic amino acid, basic amino acid. Non-standard amino acid derivative found in protein, 4-hydroxyproline and 5-hydroxylysine in collagen. You can see. Many additional non-standard amino acids are found in cell but not in protein. Ornithine and cytidine, intermediate in amino acid metabolism. Essential and non-essential amino acid. Essential amino acid, indispensable amino acid, cannot be synthesized by human, must be supplied in diet. Semi-essential amino acid required by infant and growing children. Amino acid has both a basic amino groups and acidic carboxylic acid groups. In neutral solution, the amino acid contains a negatively charged and positive charge. It is called a Twitter, German for habitat. Amino acid ionized to various state depends on pH value, at which amino acid has equal positive and negative charge, no net electric charge. So you can see the Twitter. So this is the pK value. Now friend, peptides. What is the peptides? Peptide bond, the special name given to the amide bond between the carboxyl group of one amino acid and the amino group of another. So characteristic of peptide bond. Peptide bond have partial double bond feature. You can see a rigid and unable to rotate freely. So peptide bond is planar, transconfiguration and uncharged. Peptide plane, the 6 atoms attached to the peptide bond are covalent. The carbonyl oxygens and the amide by hydrogens are in transposition. So peptide chain is directional. Amino terminal or N-terminal, the end having a free amino groups. Carboxyl terminal or carbon terminal, the end having free carboxyl group. You can see in this picture N-terminal and C-terminal. By convention, N-terminal is taken as the beginning of the peptide chain and put to the left, C-terminal at the right. So, polymers of amino acid, peptide can be classified according to how many amino acids they contains: dipeptide, tripeptide, oligopeptide, and polypeptide. Now, glutathione, tripeptide, glutamic acid, cysteine, and glycine functions important biological oxidation reduction reactions has reduced and oxidized form. The most important molecules we need to stay healthy and prevent disease. You can see glutamic acid reduced from GSC. You can see the cysteine, glycine, so oxidized from glutathione disulfide. So the key point, general structure I discuss here: alpha amino acid, chiral DL form of alpha amino acid, and classification of amino acid: non-polar, uncharged, polar, aromatic, acidic, and basic amino acid. Essential amino acid: deuterion, and characteristic of peptide bond and glutathione structure and function. So I hope, my friends, this video will be helpful to you. If you like this video, then subscribe my channel, and thanks for watching.